For Chapter 9, Biotechnology, Part 1. In Chapter 1, I mentioned that the vast majority of microbes are beneficial or harmless to us, and I went through all the different types of benefits that we derive from the microbes. And in general, they were in three groups: ecological benefits that benefit life on Earth, personal benefits, benefits you get from your own microbiota that live on and in your body, and the last one was biotechnology. The basic definition of biotechnology is using organisms, and in this case, microorganisms, to get a product that we want. So manipulating the microbes to get them to produce something we want. And the most obvious example of that is food. And within that, yeast, which is a unicellular fungi. If we introduce it to flour and water, we will get bread. If we change the conditions a little bit, we can get alcohol. And there are all sorts of bacteria and fungi that give us different types of products, usually as a result from fermentation. Genetic engineering is a type of biotechnology, and this is actually probably what you are thinking of when you hear the term biotechnology. But genetic engineering is taking into account gene expression. And remember gene expression, in gene expression, genes are composed of DNA. And remember, a gene is a coding segment of DNA, and it encodes the information to produce some sort of protein. So that is the basic idea behind gene expression, behind the central dogma of biology. And genetic engineering basically means that individuals are engineering the genes so that we can get a protein product that we want. So we are still manipulating the organisms, specifically the microbes, and this time we're manipulating their genes so that we can get the protein product. This is sort of a summary of what happens during genetic engineering. And in this case, first thing we have to do is we have to find the gene that produces the protein we want. So often the protein we want is from a human cell or a virus or some other microorganism. So we're looking for the protein that we want. And in order to get the information to make that protein, we have to find the gene. So we call that the gene of interest or the DNA of interest. And so what we need to do is cut that gene out of the genome from the original cell. Then what happens is we insert that gene into a vector. And often the vector is a plasmid because plasmids are very easy to work with. Remember, plasmids are small circular double-stranded DNA that you can find in bacterial cells. So what the scientists do is they insert that gene of interest, and it could be a human gene, into a bacterial plasmid. Then they transform bacteria with that human gene, because bacteria have the ability to pick up naked DNA, that's transformation, even when that naked DNA is just a plasmid that is mixed with the bacteria. Once the bacteria pick up that plasmid, then they start expressing that gene. And so then the protein production of that gene, that protein can be purified and it can be used for therapeutic agents, like different types of protein hormones can be used for treatment or even vaccines. Also, you can use that protein to transform other organisms, so plants or animals, to produce a particular protein of interest. And then also these bacteria can be used, you can grow them up in large quantities to actually study the sequence of DNA of that particular protein. So this is the basics of genetic engineering. When I talked about the history of microbiology, I mentioned that you need to know the year 1953. And 1953 is very important for anybody studying biology. This was when Watson and Crick made their discovery, and their discovery was the structure of DNA. The fact that DNA is a double-stranded helix and that it is arranged by complementary base pairing. And so it was 1953 when they discovered the structure of DNA. And that wasn't really that long ago. It's about a lifetime, a person's lifetime ago. It was less than 70 years ago. Before that, we had no idea of even the structure of the DNA, let alone transcription and translation, or how to manipulate DNA. So in a very short time, technology has improved so rapidly that now we are able to manipulate the genes of microorganisms, mammals, and even people. I just want to point out that there are a lot of genetically engineered products that we use every day. And this table just gives you some examples of some of the pharmaceutical products, things we use on humans to treat different disorders. And just a couple of things, cervical cancer vaccine, so that's the HPV vaccine, that is made from genetically engineered bacteria. Also, a hepatitis vaccine is made this way. Other therapeutic factors like human growth hormone and human insulin are actually made by genetically engineered microbes. And again, some more pharmaceutical products that are produced by genetically engineered microbes. Influenza vaccine can be made this way. Interleukins, which are used to treat different types of cancers and immune disorders. Also, there is Taxol, which is a cancer treatment, and other types of molecules that we use to treat different types of diseases. You should also be aware that genetically engineered products are involved in agriculture. So with agriculture, you have a couple different areas. This area gives you examples of genetic engineering in plants. And often it is to make the plants pesticides for the plants, or herbicides so they are resistant to weeds, or even to improve their productivity. And animals can be treated with genetically engineered products such as growth hormone for the cows and growth hormone for the pigs, or the animals themselves can actually have their genes manipulated so that they can produce more products or modified products like with medicine in their milk. So that's all experimental at this point. Also, food can be produced with genetically engineered products. Rennin is an enzyme that causes milk curd formation, which is involved in cheese production. So genetic engineering is very, very involved in producing food in many different ways. The process of genetic engineering involves recombinant DNA. And recombinant DNA is when a scientist makes a new combination of DNA that does not exist in nature. And for this example, remember in genetic engineering, you are taking a gene from one organism and putting it into the bacteria to make that protein product. So in this case, you may want to produce human growth hormone for treatment of individuals who need that. So human growth hormone, the gene is in the human cell. So the first thing you would have to do is cut that gene out. So here is the gene for human growth hormone, and then you would want to put it into a plasmid. So this is the blue plasmid. And in order to do that, you would need to cut the plasmid and then insert the gene into the plasmid. And after you have done that, inserted the gene, then you have created recombinant DNA. So this is a DNA molecule that is composed of a bacterial plasmid and a human gene. So this is a type of DNA that does not exist in nature. It is a new combination of DNA. It is recombinant DNA. And then this DNA can be used to transform bacteria, and then those bacteria can produce the human growth hormone that you can purify and use for therapy for people who need that. When you think about the word engineering, a basic definition could be the process of designing and building something. So when we're talking about genetic engineering, we're talking about the process of designing and building molecules of DNA, and specifically that recombinant DNA. And in order to do this, you need to be able to cut the DNA. So cut out the gene of interest and cut the plasmid that you're going to put that gene into. Once you have cut the DNA, you need some sort of way to glue it together. So you need a way to form the covalent bonds so that that gene will become a permanent part of the plasmid. And that is what makes the recombinant DNA, the new combination of DNA that does not exist in nature. And often what genetic engineers want to do once they have formed that recombinant DNA is they want to make copies of it so that they have lots of copies of the recombinant DNA so they can use it for transformation or they can study it. So these are sort of the basic tools that you would need to manipulate the DNA to make recombinant DNA during genetic engineering. And these tools that the scientists use are not little mini scissors or little bottles of glue or copy machines. These tools are actually naturally occurring enzymes. So the tools that are used to manipulate the DNA are actually enzymes that are found in nature that already have evolved to cut DNA, glue DNA, and make copies of DNA. So what I want to do now is go through several different types of these tools, these enzymes, and discuss how genetic engineers use them to manipulate the DNA. Two of these tools, or enzymes, I've already talked about in the previous lecture, and one is DNA polymerase. And if you remember, DNA polymerase is an enzyme that makes polymers of DNA. So DNA polymerase will bind to a template strand of DNA and it will make a complementary copy. Remember complementary base pairing where adenine lines up across from thymine and guanine lines up across from cytosine. So that is the DNA polymerase. And I talked about with DNA replication. All cells are capable of replicating their DNA. And remember, they do that right before they divide. So all cells have DNA polymerases, and the job of the DNA polymerase is to make an identical copy of the original strand of DNA. So scientists have determined this, that that's what DNA polymerase can do, and so we use DNA polymerase to make copies of DNA that we're interested in. The other enzyme that relates to DNA replication is ligase. And if you remember, ligase is the enzyme that forms the covalent bonds in the sugar phosphate backbone of DNA molecules. So remember, during DNA replication, one strand was the leading strand that had continuous replication because the enzymes move in only one direction. The other strand was discontinuous replication, or it's called the lagging strand, and it could only make complementary DNA in this way. So these sort of backstitching, they call it. So the ligase is needed to form those covalent bonds in that newly synthesized sugar phosphate backbone. So ligase is sort of like the glue that we can use to form those covalent bonds in the sugar phosphate backbone to make a permanent recombinant DNA structure. Restriction enzymes are a type of enzyme that I have not talked about before, and the purpose of a restriction enzyme or a restriction endonuclease is to cut DNA. So that is their job, to cut DNA into different fragments. And restriction enzymes are naturally occurring bacterial enzymes. So most bacteria have many different types of restriction enzymes. And the purpose of these restriction enzymes is to produce or to protect bacteria from phage infection. So if you remember bacteriophages have a complex capsid, and their genome is double-stranded circular DNA, even though this doesn't look circular here. So double-stranded circular DNA. And remember, after attachment, when they use their tail fibers to recognize and attach to the appropriate host cell, then they penetrate by injection. So they inject the DNA genome into the bacterial cell. And then usually what would happen in the phage life cycle is that the host DNA would get chopped up into little pieces, and then the phage DNA would take over control of the cell to go through biosynthesis, maturation, and ultimately release, which would kill the cell. Well, a restriction enzyme is the way the bacterial cell protects itself from phage infection. Because as soon as this phage DNA is injected into the host cell, the restriction enzymes will bind to that DNA and chop that DNA up into little pieces. So it's degrading the phage DNA into small fragments, and that protects the bacterial cell from infection. So that basically stops the phage life cycle. So these restriction enzymes evolved in nature to protect bacteria from phage infection by cutting the phage DNA into small fragments. Restriction enzymes are naturally occurring bacterial enzymes that cut DNA. And the reason we refer to them as restriction enzymes is that each enzyme will only cut at a certain sequence of DNA. So for instance, this restriction enzyme will only cut at this sequence of DNA. So it is restricted to cutting the DNA only when it comes upon DNA with that sequence. And when it cuts the DNA, what we are actually referring to is it is breaking the covalent bonds in the sugar phosphate backbone. So when it cuts the DNA, it's actually breaking covalent bonds in the sugar phosphate backbone, and most of the time, restriction enzymes cut in a staggered manner. So they're not cutting straight across the DNA, they're cutting in a staggered manner, and that creates sticky ends. And when we refer to them as sticky ends, what that means is that this overhang, like in this case, GATC, that overhang, if it comes in contact with a complementary strand, which would be CTAG, it will stick to it, it will hydrogen bond to it. So that's what we mean by sticky ends. So if that end comes in, that overhang on that end comes in contact with a complementary sequence, it will hydrogen bond very quickly to it, it will stick to it. There are actually hundreds of different types of restriction enzymes or restriction endonucleases. And all of these enzymes are used in recombinant DNA technology. So that little R DNA, R stands for recombinant. And these are just an example of 4 different common types of restriction enzymes. And probably the most famous one is EcoR1. EcoR1 comes from E. coli, so that's the eco part, and it's restriction enzyme 1. So this was the first one that was found. And for all these enzymes, this shows you what the restriction site is. So EcoR1 will always cut at this restriction site. And if you look at this restriction site, this is actually a palindrome. And a palindrome in English is a word that is spelled the same forwards and backwards, like names like Otto or Anna. When you refer to a palindrome in DNA, what that means is that one side of the double helix is the same as the other side going the other way. So for this, the top strand of this double helix is GAATTC. If you go the other direction on the complementary strand, it has the same exact sequence, GAATTC. So all restriction enzymes cut at palindromes, so DNA palindromes. And again, most of them cut in a staggered way, so EcoR1 will break that covalent bond, and then it will break this covalent bond. So then what you notice is the 2 sticky ends are complementary to each other. So this is EcoR1, and again, every restriction enzyme has its own unique recognition sequence. HindD3, this is from Haemophilus influenzae, and it is the 3rd enzyme they identified, and HindD3 will only cut at this sequence, which is a different sequence than EcoR1, of course. The fact that restriction enzymes can cut DNA and leave sticky ends is very important for forming recombinant DNA. And this figure shows you how using DNA from 2 different sources and cutting them with the same restriction enzymes makes it very easy to form recombinant DNA. So in this case, this purple DNA could be human DNA, and maybe the gene you're interested in here is human growth hormone, human insulin, or something. Some gene you are interested there. And on either side of the gene, you have an EcoR1 site. So you use the enzyme EcoR1 to cut this gene out of the human genome. So you've cut the gene out of the human genome. Then, while you're doing that, you also have this pink DNA, which could be a plasmid. So that could be a plasmid, and you are going to cut that plasmid with EcoR1 so that you are creating complementary sticky ends. Once you have cut the human gene with EcoR1 and you've cut the plasmid with EcoR1, all you have to do is mix those two different types of DNA together, and they will spontaneously stick together. Because remember, those cuts are producing complementary sticky ends. So the two genes will automatically stick together, and even this side will stick or bind to that other side, and that will create a circular piece of recombinant DNA. And at this point, it's only held together by those hydrogen bonds holding those two complementary sticky ends together. And if you remember, hydrogen bonds individually are not very strong, but you get a lot of them together, you get a strong force. But another thing about hydrogen bonds is they're also easily broken. They're easily made and easily broken. So to make sure that this is a permanent construct, a permanent recombinant DNA molecule, you need to add DNA ligase, because DNA ligase is going to fill in or form those covalent bonds in the sugar phosphate backbone. So you need to add the DNA ligase to make this a permanent molecule of recombinant DNA. And this is how you create or add a gene from another organism like a human into a bacterial plasmid. While there are hundreds of different types of enzymes that are involved in genetic engineering, I just want you to know one more, and it is reverse transcriptase, which I have talked about before. If you remember, reverse transcriptase is part of the structure of a retrovirus. And a retrovirus is an enveloped virus, and in the envelope you have those spikes that are used for attachment. Also, it's a virus, so it must have a capsid, that protein coat that protects the nucleic acid, which is RNA, for a retrovirus. And the unique thing about the retrovirus is it also carries the enzyme reverse transcriptase. And if you remember, reverse transcriptase is an enzyme that does the reverse of transcription. So what that means is it takes the RNA genome of the virus and makes a copy in DNA. So it copies that RNA into a DNA form. And reverse transcriptase is often used in genetic engineering, especially by scientists who want to study RNA, because RNA is relatively unstable. And if you're a scientist and you're studying different types of mRNAs, they break down very easily. So it's much easier for a scientist to convert that mRNA into a DNA copy because it's much easier to work with. But more recently, reverse transcriptase is really important in testing for COVID-19. So one type of test that they use to identify if somebody is positive for COVID-19 is the nucleic acid detection. And in this case, they are actually looking for RNA from the virus. So they're actually looking for the viral RNA because coronavirus is an RNA virus. So they take a swab from the person's respiratory system, and then they extract the RNA. And again, because RNA is difficult to work with, right away they want to convert it to DNA. And how do they do this? They use reverse transcriptase. So the reverse transcriptase will convert the coronavirus RNA into DNA. Then they amplify, they make copies of that DNA so that they can detect it in the test. So this is just one example of how reverse transcriptase is actually used today. In addition to the enzymes that are used as tools to manipulate DNA, electrophoresis is a process that is also often involved in genetic engineering. And the purpose of electrophoresis is to separate DNA molecules by size. So that is the purpose of this process. So often when DNA is cut with a restriction endonuclease or a restriction enzyme, you end up with a sample of DNA with different sized fragments. And it becomes important to separate out specific fragments or to organize the fragments by size. And that's what electrophoresis does. And for electrophoresis, there are two important parts. The first is electricity. So that's the electro part of the word. Electricity is going to move the DNA through a gel. So those are the two parts of electrophoresis. You need to have electricity and you need some sort of gel that the DNA is going to move through. For this process, this is a figure, and this basically goes through an example. So here is a sample of DNA, and it is cut at these three different places by a particular restriction enzyme. So now you end up with a sample of DNA that has 4 different fragments. And to organize these fragments by size, you will take the solution of DNA and you will load it into a gel. So this is an example of a gel. And these gels are often made of agarose, so the same solidifying agent that's found in the media. So you load the DNA into this well and then run electricity through the gel. So you run the electricity through the gel, and because DNA has a negative charge, the DNA is going to be moved towards the positive node, so the DNA will move through the gel. And smaller fragments are able to move more quickly, larger fragments move more slowly. So in this example, with these 4 different types of fragments, the largest fragment is going to move more slowly, so the band that has the largest fragment will be there. The smallest fragment will move more quickly, so the band that contains that fragment will be there. So this is a way to separate the different fragments of DNA by size. And this is an example of an actual electrophoresis gel. And often in these gels, you have a size marker, which is a sample of DNA of known lengths. And so as you can see, the fragments of DNA that are larger, that's about 5,000 base pairs long, is going to move much more slowly than the shorter fragment that's about 1,000 base pairs long. So this is the process of electrophoresis.